genetically modified crops. What are GM crops that we had already studied on page number 6. There we had seen that to import any GM crop we need permission of FSSAI and up to 1% sample. If it comes by mistake of GM crop, then that is allowed, right? That we have already seen. Now what is the update? The update is about genetically modified soya meal. Actually, soya meal is not genetically modified. See, what happens is, when you grow soya bean, then soya bean could be GM crop, right? Not in India, maybe outside India, the soya bean you have grown is, maybe it is GM crop, genetically modified. When you crush it, you get 20% oil and 80% the dry cake, which is called as soya meal. Now, even if your soya bean was genetically modified, but after crushing what you are getting is not considered as genetically modified. So if you are going to import soya bean, then you need to declare that the soya bean that I am importing is not a GM crop. I am importing natural soya bean. But if you are importing soya oil or soya meal, then no permission is required, because government does not consider them as GM crop. So India has been importing soya oil made from genetically modified soybean since a long time. Now we are also going to import soya meal, which has originally been made from genetically modified soybean. But since we are importing the soya meal, we are not importing the genetically modified soybean. We are importing just the soya meal. So therefore it is allowed, though we are doing it for the first time. Please remember that BT cotton is the only GM crop which is allowed for commercial cultivation in India. Next topic is about the genome mapping in Indian Ocean region. See, what is genome mapping? That we had already studied on page number 1. There we saw that genome is a complete set of DNA. It is a complete book, right? If you study each and every word individually, if you study the complete book completely, then that is your genome sequencing. However, if you make an index of the book, then that index is like a genome mapping. There you only see broadly that which genes are where. You don't go into much the details. That is genome mapping. Similarly, from DNA we get mRNA, from mRNA we get proteins. So just like genome is a complete set of DNA, proteome is a complete set of protein. That also we had seen on page number 5. Okay, so now what is the news? That National Institute of Oceanography, which is under Ministry of Science and Tech, because it is a CSIR lab, it is going to start a trace biome project. Under trace biome project, a ship will go in the Indian Ocean region. Okay, there it will drop down a cord like this. Okay, at the bottom of this cord there will be a bottle. In this bottle it will collect the water that is here. Now this bottle will be brought upwards. Now here the water in this bottle will be analyzed, because that water will have many microbes. So now scientists will do the genome and proteome mapping of those microbes in this water, right? Two techniques will be used, NGS and Sanger sequencing. NGS or next generation sequencing we had already seen on page number 6. So here what actually happens is, suppose there is this long piece of DNA. So NGS machine will simultaneously study different parts of the DNA. So in very short period of time, it will be able to make the complete genome sequencing of the entire DNA. Whereas in Sanger technique, what we do is we study the whole long DNA step by step. We will start from here and slowly we will keep on studying the DNA. So NGS is very fast as compared to Sanger, but the aim of both is same. The aim of both is genome sequencing or genome mapping. So under trace biome project, many things will be studied besides their proteome and genome mapping. Scientists will say that how the metals or minerals in the water are impacting the bacteria there. How can we use these bacteria and microbes in our medicines or in our industry, and many such things. Please remember one thing. National Institute of Oceanography is under Ministry of Science and Tech. However, in environment page number 4, we had seen 4 other institutes on ocean technology. That is National Center for Ocean Info Service, National Center for Ocean Research, Coastal Research, Ocean Technology. All these things come under Ministry of Earth Sciences, Ministry of Earth Sciences, whereas National Institute of Oceanography comes under Ministry of Science and Tech. Gene editing. So gene editing we had already studied on page number 3, where we had seen that genome is your book, and if you edit any sentence, then that is gene editing. Either you can make some correction here, or you can bring some genes or words from some another species also, right? All that is part of your gene editing. Now where to make the correction in the book? Someone needs to guide you that it's a fat book, where exactly you need to make the correction. That guide is one part. Another part is when you have identified where to make the correction, then you will need some pencil to actually make that correction, right? So that we had seen through CRISPR-Cas9, where we saw that CRISPR guides. CRISPR is a guide and Cas9 is a pencil or the scissor that actually does the cutting, right? This all we had seen. So what is the update? Update is nothing, just a small term. The term is engineered nuclease. So it is this enzyme which we are calling as engineered nuclease, which has two parts. One part is the guide, that is a targeting part, and another is a scissor, just like CRISPR-Cas9. So these enzymes or engineered nuclease can be of two types. Either they can be RNA based, which use RNA, like CRISPR-Cas9 uses RNA for gene editing, or they can be protein based, which uses protein to do this work. So engineered nuclease can be RNA based or it can be protein based. Protein based like Talens and zinc finger nuclease. So these are some techniques or enzymes to do the gene editing, and all of them, engineered nuclease, have two parts. One is a guide and another is a scissor. Department of Biotech supported the first CAR cell therapy done in India. So CAR cell therapy is generally used for treating cancer. It is basically a gene editing technology, right? But it is very expensive. It ranges from 3 to 4 crore rupees per therapy per person. So therefore it is being tried that some changes could be made in this therapy to bring down the cost. So now we need to study what is CAR cell therapy for the prelims examination. So what happens in CAR cell therapy is that, see, this we have studied earlier, that T cells are our white blood cells. They are a part of the immune system. They help the body fight the disease. So in CAR cell therapy, we take out blood. We take out blood from the patient. In that blood, there will be some T cells. We separate the T cells and we return the blood to the patient. Okay, now we have the patient's T cells in the laboratory. We are going to do gene editing on the T cells. How? We will send a harmless virus along with some specific genes. When this virus will deliver that specific gene to the T cell, then those genes will help T cells grow some special type of receptors on its outer surface. Okay, so we have inserted some genes into the T cells so that they grow some specific receptors. These T cells, now these CAR cells, now they are called as CAR cells. These CAR cells we will multiply in the laboratory into millions of cells. Now we have millions of CAR cells, we will insert them back into the patient. Now the receptors on the CAR cells will find the cancer cell. They will direct the T cell to the cancer cell, they will attach to it and they will destroy the cancer cell. So that is how we are using CAR cell therapy to destroy cancer. So what does CAR stand for? In CAR cell, CAR stands for chimeric antigen receptor. It is a type of immunotherapy. Immunotherapy means strengthening our body's immune system to fight some disease. Here we take the T cells from the patient or from a donor. We modify the T cell in the lab. We multiply them into the millions. Then we insert them back into the patient. There they will attack and kill the cancer cell. So here the CAR cells are acting like a living drug. UPSC can simply ask, what are Yescarta, Kymria or Tecartus? They are types of CAR cell therapy. Next topic is about the RNA interference. RNA interference, previous year questions were quite simple and straightforward. We had already discussed them on page number 6. Now there was some news that RNA interference technology can be used to make pest resistant plants, and the same thing is given in your NCERT class 12 book also. So let us see this thing in a little bit detail, but don't worry, it is quite simple. So first let's understand what is the meaning of RNA interference. See, look at this diagram. This is a human cell. We are talking about humans, right? So what happens naturally is that in our cell there is nucleus. In nucleus there is DNA, which has our genetic information. From DNA, mRNA is made, and through the process of translation, mRNA is converted into proteins through which our body is made. This we had understood earlier. Now what happens is that suppose, okay, don't look at this photo right now. Don't look at this locust. Now suppose there is a virus outside. That virus entered our cell. Virus will have some RNA, just like your corona. Okay. Now that RNA has entered our cell here. Now our ribosome will use this foreign RNA to make proteins or more viruses as per the virus. But that is wrong. So what our body will do is it will naturally interfere in this process. Our body, see, what will happen is that this foreign RNA will get converted through translation into some protein. Our body will naturally interfere in this process of translation. That is called as RNA interference. So RNA interference is a natural process for gene silencing. You are silencing the genes of this foreign virus. It is a part of our immune response. It prevents the manufacture of some specific proteins, because proteins are made when mRNA is translated into proteins, right? So that is a natural process. Now what we are doing is we are using this natural process in such a way that our plants won't be affected by some pest like locust. How we are doing that? Now suppose this cell is of this locust. So in the locust there is a nucleus. It will have DNA of the locust. Then mRNA will be formed, and from mRNA proteins will be formed. From some protein the legs of the locust will be formed. From some protein the wings of the locust will be formed, and so on, right? Now what we are going to do is, we know that locust is going to eat the plants. On the plant we will spray some chemicals. When the locust will eat the plant, the chemicals will go inside the body of the locust. In those chemicals there will be some RNA. That RNA will enter the cells of the locust. Now what this RNA is going to do, what these chemicals are going to do, they will interfere in the natural process through which locust is made. Okay, it is a bit different process here. What we are saying is that in the locust body, through locust DNA we got locust RNA. From that RNA, body of the locust is formed. The chemicals that we have inserted into the body of the locust, those chemicals will interfere in this natural process of protein formation in the locust's body. So maybe now we are saying that the wings of the locust will no longer be formed, or the legs of the locust will no longer be formed. Right? When the legs or wings are not formed, then the locust is of no use. It will not be go from one place to another and destroy the crops. So hence we are able to make our plants as pest resistant. So this is one of the ways. More than this, I don't think UPSC will ask. Even if it asks, then just listen to some terms, like there is something called as dsRNA. RNA is almost always single stranded, but sometimes the RNA can be double stranded like this. Okay, what is dicer? Dicer is an enzyme which dices the long dsRNA into small parts like this. Then out of this small double stranded RNA, Okay, two parts will be done and it will be made into single stranded RNA, a small single stranded RNA, that is ssRNA. Now in the risk complex, what will happen that there is a naturally long mRNA, and through the risk complex this small portion is going to stick to the mRNA, and therefore the whole of mRNA will not be able to convert into the proteins. That is, we are interfering in a way, we are interfering in the conversion of RNA into the protein finally. If you understood this, very good. If you did not understand, don't worry, UPSC will not ask in such a detail. Just remember that these terms dsRNA, dicer, risk, these are related to the RNA interference technology. To understand blockchain, first understand what is ledger. Ledger is nothing but a record of all the transactions. Three people have a bank account. Bank maintains their ledgers. Bank maintains how much money came and how much money went out, so how much money is left in the account. So that is a centralized ledger. Problem is that if the bank is corrupt, then the bank will do some fake transactions, or he will do some changes in the ledger such that you will lose your money. So that is a problem with centralized ledger. Solution is you can have a distributed ledger. Suppose the bank says that whatever transactions happens in the bank with any customer, all the records, all the transactions will be shown to everyone, and everyone will keep on recording them in their own ledgers. Right? Everyone will maintain the ledgers. Bank will tell everyone that these transactions are happening, information is shared, and everyone is maintaining the ledger. So tomorrow if the bank goes corrupt and bank changes the ledger which it has, then other people can say no, our transactions are matching, it is you who is doing something wrong. Right? So that is a benefit of distributed ledger. Google Docs is an example of distributed ledger. You can make a Google Doc and you can share it with your friends. You can also make the changes, others can also make the changes. So in a way, whatever transactions are happening, you can make a proper record in the Google Docs. Right? Now the problem here is that tomorrow one of your friends can make changes in the Google Docs. Some transaction happened, but he can edit it, so it is editable. Right? So one solution is, instead of Google Docs you can use SMS chat. So in SMS chat you also have the record and the other person also has the record. You both have the records of SMS chat, but once an SMS is sent, you cannot edit it. Right? It is non-editable. So SMS chat and Google Docs both are shared, both are like a distributed ledger. The difference is that in SMS chat you cannot edit it, but in Google Docs you can edit it. So what is blockchain? Blockchain is a combination of two. Blockchain is a distributed ledger which is non-editable, just like a SMS chat. Fine. Now there are many applications of blockchain. There can be application of bank chain, cryptocurrency, smart contract, non-fungible token. All these are different applications of blockchain technology. So what happens is, like in case of bank chain, in 2017 State Bank of India launched bank chain in India. It said that in some cases, it is a pilot project, okay, in some cases when KYC of a customer is obtained by a bank, by this bank, or some big corporate has, he has given his property to the bank, the papers of the property to the bank, and taken a fat loan from him, from the bank, then the bank is going to share these information with other banks also, and these banks will also share their information with each other. Right? So that if this businessman makes some fake papers of property and goes to another bank and shows those papers and tries to get the loan, then this bank will deny, because this bank will say that we already know that you have submitted the property papers to this bank, so you are trying to do a fraud. Right? So there are many applications. So it is being experimented that how we can use blockchain technology in banking system to reduce the frauds. That is one application. Another application is about the cryptocurrency. So when the transaction happens, you send bitcoins from one person to another, then suppose on a piece of paper you write down that who has given money to which person. Right? You keep on recording the transactions. When there are so many transactions, you combine them into a bundle of papers. Then you combine the bundle of papers into a box, and you name this box. You name this box as hash 1234. So the blocks, it is a block, right? The blocks are identified through the hash. Now you send these blocks to different people in a distributed ledger. Different people are keeping a record, right? So it is a record. Now you send this record to different people. All of them will keep it in their record, right? So this particular person, he once got a block, he has kept it here. Next he will get the next block, then it will get the next block. So in a way, a chain of blocks is made, just like a chain of SMS is made which cannot be edited. Similarly, a chain of blocks is made which cannot be edited. So this is called as a blockchain, which is used for cryptocurrency, right? So cryptocurrency is a type of virtual currency. Rupee is a real currency. Rupee is a real currency. You can hold the rupee in your hand, or you can store it in your bank, in your Paytm wallet, anywhere. But rupee is not a virtual currency. Rupee is a real currency. By December 2021, RBI may launch a virtual currency. Virtual currency is just like your rupee. RBI will issue it, but it will not be in the physical form. It will only be in the digital form. Okay. Now problem is that when people are making fake notes, right? They have printer at home, they will print the fake notes. So similarly, people can create fake virtual currency also, right? Even in fact, creating virtual currency fake is quite easier, right? So you need to secure it. One way of securing the virtual currency is to use cryptography. In such a way, we will call such a currency as cryptocurrency if it is using cryptography to secure the virtual currency. Now there are different techniques of using cryptography. One of the techniques of using cryptography is blockchain, right? So in blockchain, you are securely storing the data. It is a type of cryptography, and it is just one type of cryptography. So if you are using blockchain technology to do cryptography to secure a virtual currency, then it is called as cryptocurrency. Fine. So cryptocurrencies they are not issued by any central bank. Central bank may issue them. But generally central banks do not issue the cryptocurrencies. Anyone with a computer can mine it. How much money supply will be there, that RBI decides. How much new notes to print, that RBI decides. But if you have a computer and internet connection and lots of time, then you can create cryptocurrency. You don't need a central bank for that. That is how the cryptocurrencies are created, right? And they have same value across the world. So rupee dollar exchange rate changes every day, but the value of Bitcoin, what it is in India, the same value of Bitcoin is in USA. Bitcoin is same whether it is in India or it is in USA, right? Problem is, the two people who are doing the transaction in Bitcoin, they are totally anonymous. There is no know your customer or KYC in the cryptocurrency. Central banks may say that if you're doing something, you must have KYC, else we will not allow you, and all such things. But the cryptocurrency concept in itself does not require you any KYC or anything such thing. It simply says that your anonymity is totally maintained. Just like Telegram, if you have another person's Telegram ID, then you can send him the messages. There is no need to know the telephone number of that person. Same is the thing. There is total anonymity. Problem is that each time an authentication is done, each time a transaction happens and it is stored in many places, then lots of electricity is used, right? So that is one problem with it. Volatile value. Its value changes very rapidly. Bitcoin today is worth something and tomorrow Bitcoin may be worth some other thing, because its value is not backed by any central bank. Money supply, right? If so many people start creating cryptocurrency Bitcoin, then there will be so many cryptocurrency in the world. But rupee that RBI controls. Some examples of cryptocurrencies are Bitcoin, Ethereum, Ripple, Litecoin, and Tether. See the 2016 question on Bitcoin. Bitcoins are tracked by RBI? No, RBI cannot track the Bitcoins transactions. They are totally anonymous. Anyone can with a Bitcoin address can send and receive Bitcoins. Yes, just like Telegram, without knowing the identity of the other person. So 2 and 3 are correct. 2020 question on blockchain technology. Blockchain is a public ledger that everyone can inspect but no one controls it. Yes, here we saw that blockchain is a public ledger. It means many people are having the records and they can inspect it, but there is no single party which will be able to modify or control it. So first is correct. Second says that the blockchain is designed in such a way that only cryptocurrency you can make. No, through blockchain you can make bank chain, you can make smart contract, you can make non-fungible token, many things you can make. Second is wrong. Third is that applications that depend on basic feature of blockchain can be developed without anybody's permission. Yes, when Bitcoin was made, then you did not take permission of RBI or US Fed. Bitcoin was made if you have sufficient participants. If you have sufficient participants, then it will work, right? So third is also correct. Smart contracts. Smart contract is quite simple. Initially you tell the some conditions which need to be met. For example, in the share market you tell that if the share of such a company comes to this particular value, then my trade should be executed. I want to buy or sell at this particular value. You are telling the exchange, right? Then so many people are telling the exchange such a thing, that I want to buy or sell at such a price, such a price, such a price. Now the computer is continuously matching the bids, and the moment the bids are matched, the contract will be executed by itself, and you don't know who the other party is, right? Everything is happening by itself. So such a things are called as smart contracts. They are now being secured. It is being tried that we should have such smart contracts between two people, where the details of the contracts are written, conditions are written, and computer will keep a track. Whenever the conditions are met, the contract will be executed automatically, and it will be secured through blockchain technology, and some central government authority will keep a track on such transactions. That is how blockchain technology is being designed. This is another application. Non-fungible token. देखो, non-fungible token. First understand what is the meaning of fungible. Fungibility means replaceability. You have a 100 rupee note, but it is quite old now. It doesn't look good. So you exchange the 100 rupee note with your friend who has a brand new note. Can you do this? Yes, you can do. It is 100 rupee note only, na? So you can always exchange it. So these are replaceable. These are fungible. But if you have the original Mona Lisa painting, okay, over time painting's quality reduces. It becomes a bit fade. So you will think that now the Mona Lisa, I have the original painting, but it is now old, so I'm not liking it. You print a new painting from the printer, and that is looking very nice. So you replace the original painting with the new painting. Can you do this? No, you cannot do this, right? So the real art, the painting which was originally made, it is non-replaceable. It is non-fungible, right? Now, how do you authenticate? How will you say that the painting that I am having is the original one and this is the fake one? How do you differentiate? To differentiate, you will put a tag. You will take a tag and you will staple a tag with the original painting saying that this is a real painting and all other paintings will be the replica of this. They will be fake ones. Now what the thief will do, bad people will create duplicate tags, right? They will also put the tag on their painting and say that this is the original one. So what is the solution? Solution is you use blockchain technology to certify the authenticity. So what we are doing is, in blockchain we store some data at many places, right? So will you store the original painting in many places? No, you are not storing the original painting in many places. Original painting, suppose it is a digital painting, so it is stored only in one place. The original painting will be stored only in the one place only, right? But the token that is the certificate that is saying that this is the original thing, that token's authenticity will be confirmed through the blockchain technology. So please remember, here we are using a token through blockchain technology which is certifying the authenticity of a digital good. Although Mona Lisa painting is not a digital good, I'm just giving an example of Mona Lisa painting. Otherwise, this non-fungible token is generally now used to prove the authenticity of some digital goods. So fungibility means it is replaceable by another identical item like a note, currency notes. Non-fungible means it cannot be replaced by another identical item like your paintings or some digital goods. Non-fungible token is a proof that the product is original. It is a non-fungible token is a piece of data that certifies that the digital asset you have is unique and original. So this is the fifth class of Science and Tech. In this fifth class we have studied some pages here, and rest of the pages are a part of our paid course. So what happens is, in each class the first few pages are explained on the YouTube and the rest of the pages are explained in our paid course. Okay, please remember we are talking about the explanation video only. We are talking about the explanation video. The PDF is totally free. The PDF of entire course is totally free. It is available on our website. So in every class some portion is explained on the YouTube and remaining portion is explained in the paid course. The cost of the paid course is 200 rupees for Hindi video and 250 rupees for English video. Like this is English video. See, PDF is always in English only. Okay, we are talking about the explanation voice. Here my voice is only in English, so 250 rupees for this course. If you are okay with English explanation in which Hindi and English both are used, then you can pay just 200 rupees and purchase this course. How to purchase the course? You can purchase our course either on the mobile app or on the laptop. Mobile app, the descriptions is given in the description box of this video. Their links are given. Click on those links and you will be able to download the mobile app either through the Play Store or through the Apple Store. If you are using Apple mobiles, then please, after installing the application, please enter organization code as MXLDF. So if you are using mobile app, then open the mobile app, click on store at the bottom of the screen. In the store, click on the course that you want to purchase, make the payment. After payment, again open the store and you will be able to see the videos. If you want to see the videos on laptop, then the link is given in the description box. Click on the link, register yourself, make the payment. After payment, click on my courses. You will be able to see the videos. Please note that you should make the payment only on the platform where you want to see the videos. If you are making the payment on the laptop, then you will not be able to see the video on the mobile app. Similarly, if you make payment on the mobile app, you will not be able to see it on the laptop, right? So choose only one platform. Whenever new video will come, if you want its notification, then please subscribe to our channel and click on the bell icon. Then you will get notification of the new video.